Hello, everyone, and thank you for joining our webinar entitled Approaches to Enhance Demanding LC and SFC Chiral Separations. Today, our speaker is Dr. Daniel W. Armstrong. Dr. Armstrong is the Welch Distinguished Professor of Chemistry and Biochemistry at the University of Texas, Arlington. He has over 800 publications and received over 35 awards for teaching and research, including numerous ACS awards and the Chirality Medal. He has been named one of the top 10 analytical scientists in the world by Analytical Scientist magazine and was elected to the National Academy of Inventors in 2014. He has mentored and graduated over 120 PhD analytical chemists, nearly half of which were women. Dr. Armstrong developed the most comprehensive solvation and characterization models for room temperature ionic liquids and pioneered their use in analytical chemistry. He has also developed ILs as the most versatile MS reagents. His work and columns were in part responsible for the chromatography-led revolution in chiral separations and provided the impetus for the FDA's regulatory changes regarding chiral drug development in 1992. He has developed rapid, high-efficiency microfluidic methods for analyzing microorganisms. His work has been cited over 53,000 times, and he has a Hirsch index rating of 116. He has organized many international conferences and over 100 short courses worldwide. So before I hand it over to Dr. Armstrong, I wanted to mention that if you have questions that come up during the presentation, you can click the chat icon at the top of your screen and type them in. Once the webinar has concluded, Dr. Armstrong will address the ones that he can get to in the amount of time left. If the webinar ends and your question has not been answered, please follow up with us at sales@jascoinc.com. This email address will be listed on our final slide as well, so don't worry if you didn't catch it the first time. So, without further ado, I would like to welcome Dr. Daniel W. Armstrong. I'm sorry, Dr. Armstrong, you're still muted.
There we go. Can you hear me now?
We got you. Thank you.
Okay. Thank you for that overwhelming introduction, Tom. We're going to be covering a lot of area just in chiral separations today, but there is going to be a lot of unusual things that we've been asked to do by outside researchers. And so let's get started right away. And for some reason, it's not shifting. Oh, there we go. If I click. So we deal a lot with outside chemists, and many of them are organic chemists, very talented organic chemists, and we help them on their synthesis problems and their analysis. And so it helps to understand, to interpret what they're saying in terms of analytical chemistry. So they provide samples that they say are pure by NMR. And so as an analytical chemist, you have to be able to translate this into analytical terminology. And the translation of pure by NMR is impure. So once you know that from the outset, you are not surprised quite as many times. Now, also a bit of nomenclature. Some of the enantiomers we're going to be dealing with, and in fact, a decent proportion of them are called atrope isomers. They have helical chirality. Instead of having an RS enantiomer or a DL enantiomer, they have a different terminology. They use the prefix capital P or delta for a right-handed helix, or a capital M or lambda for a left-handed helix. And the helicines provide a nice teaching case of this. You can see that the helicine on the left, if you look down the axis of chirality, it spirals in a clockwise direction. So this would be labeled the P or delta helicity or enantiomer. And the opposite enantiomer, if you look at it, it's spiraling in a counterclockwise direction. So it would have the M or lambda helicity. We'll be doing or showing a lot of different separations, probably half of them, at least half of them, will be by supercritical fluid chromatography. This is our unit, which just happens to be a JASCO unit, but we have this one because we can do both analytical and semi-prep separations, both of which we do quite a bit of in our laboratory. Now, I'm going to first introduce you to what is the newest chiral selector. Although we introduced it a few years ago, a lot of people aren't familiar with it. It's based on cyclofructans. They're actually called cyclofructan. You can see a structure of a cyclofructan here on the right, bordered in blue. It's a cyclic oligosaccharide like cyclodextrin, but it's very different from a cyclodextrin. These are beta 2,1 link fructose units, and they have no hydrophobic core, no classic hydrophobic inclusion complexes possible. What they do have is an 18 crown 6 in the middle, and they're of course chiral because they're composed of sugar units. Now, when we originally started working with cyclofructans, we thought, oh, this is going to be a great new chiral selector. So we took 250 chiral drugs, made a column of it, and it separated exactly 6 of them, which was hugely disappointing. And on the right, you see one of the better separations, which is not a very good separation. We then learned, because the cyclodextrins are folded in on themselves, you have to derivatize 6 or 7 of the hydroxyl groups. It opens up, exposes the core. Suddenly, out of the same screen, 250 compounds, we observed over 150 separations. And so I'm going to talk about a couple iterations of this and how we used it to solve problems for other researchers. One of the 2 dominant forms of cyclofructan, chiral stationary phases, is the isopropylated cyclofructan 6. It is the best chiral selector and column for separating primary amines of all kinds. And it does it with organic solvents and supercritical fluids, both. And you don't have to have a strong acid in the mobile phase. And so these are typical separations of organic amines that occur. It's done with 60% acetonitrile methanol with a few drops of acetic acid triethylamine. Out of 119 racemic amines, we had a 93% success rate of separating them. So it's almost universal. These are 2 of the more unusual types. If you look on the left, there's the primary amine group, and it has 7 atoms from the chiral center. 1, 2, 3, 4, 5, 6, 7. So the main interactive group is far from the chiral center. Usually those are difficult to impossible separations, but it's simple for the cyclofructan P. On the right-hand side, you see essentially a bicyclic hydrocarbon that has the 1 functional group, which is the primary amine, yet beautiful separation of this as well. We screened the commercial name is Lyric CF6P for this particular chiral stationary phase. We screened it with 30 primary amines against all 6 of the dicel columns. So the 6 dicel columns are in the yellow circles around the big blue circle. So it's 1 column against 6. And the CF6P for primary amines had 26 unique hits out of 30, whereas the sum total of all 6 of the dicel columns had the same number of unique hits, 26. So you can do on 1 column pretty much what you can do on 6 columns of another type. And once again, we are operating in acetonitrile methanol type mobile phases or supercritical fluids. Loading capacity is excellent on these stationary phases. On the top, you see for naphtholethylamine, analytical separation, 16 micrograms. We increase the volume to semi-prep, 3.2 milligrams. You see the chromatogram you get on the bottom. So excellent preparative properties for these things. So this was done on an analytical column of 4.6 by 25 centimeter column, a classic analytical column. But I now want to change the focus to a different derivative of cyclofructan. These are naphthoethyl functionalized, so they have aromatic functional groups on the cyclodextrin. Here you see a molecular model of it. These are called Valeric shell RN, and these were specifically designed to separate atrope isomers. that kind of lambda and delta things that we talked about before. It does do primary amines as well, and along with Lyric Shell P, they have a 98% success rate for any all primary amines, but we're going to focus on atrope isomers for this part of the talk. One of the outside people we began working with is Sherry McFarland. Sherry McFarland had a series of ruthenium and osmium complexes. So they had these bidentate ligands complexing like the central metal, which make them chiral. So they exist as delta and lambda isomers, and she needed not only analytical separations, but preparative separations for these things. And she had a few of them. She had 23 to be exact at this point. Here that you see the structure of all 23 of these compounds, which I'm not going to dwell on. They're all various iterations of ruthenium, and a few of them with osmium in the core. But there's a select few of these, particularly the first 5 that are hugely important. For the reason I'm going to tell you, this particular compound is under phase 2 clinical trials for bladder cancer. It's having some amazing results as a photoreagent for bladder cancer. But they had never separated the 2 enantiomers, and you aren't going to get it through phase 3 clinical trials in the FDA unless you've tested both enantiomers. So we had to develop the separations of these for her research group. And here are the 5 most important compounds. The therapeutic ones have these thiophene side chains, 3 or 4 thiophene side chains that really adds to therapeutic activity. And so using SFC in this case and the cyclofructan 6-RN column, you can see we can easily separate all the delta and lambda isomers. We also had to determine the absolute configuration. We did this by vibrational circular dichroism with Jordan Nafty of BioTools. And we prepped over 100 milligrams of each enantiomer of every one of these things using the JASCO SFC. And so it worked out quite nicely. And as you can see, with this kind of a separation, it's going to be an easy prep, and it was. There was one unusual thing, though, about these separations. So the mobile phase is going to be 55% carbon dioxide, and it's going to be 45% modifier. Now, the modifier you can use is, usually it's methanol, but we can use methanol or acetonitrile. And we found that a mixture of methanol and acetonitrile produced the best results, and that is shown as you see on this figure. We're plotting the retention factor on the y-axis and the organic modifier composition on the x-axis, with methanol going from 0 to 100%, and the opposite for acetonitrile. And these are the Ks, the retention factors for both of the 2 enantiomers. And what you see is the selectivity, the difference between the K, doesn't really change as you change the ratio and the organic modifiers. However, the retention factor changes tremendously. You get this great speeding up of the separation, depression of the retention factor at 65% methanol, 45% acetonitrile, with still practically the same selectivity as you had of all of the ratios. So obviously, for the analytical and particularly for the preparative separations, this is the ratio of the modifiers that we're going to be using, because it just goes a lot faster. So this was something we didn't expect that we found out during the course of these experiments. Now let's go to another scientist who had a problem. This is Mike Crochet at University of Texas, Austin. And he had synthesized a whole series of atrope isomers, mainly atrope isomers, not completely, of sulfonic acid derivatives. So they're going to be negatively charged. Strong acid, you see the sulfonate groups on all of these. And phosphoric or phosphatic acid, in this case, phosphoric acid derivatives. So the top half have sulfonate groups, the bottom half have phosphoric acid groups. And these are going to be ionized. They're going to be negatively charged. And Mike usually comes to us when he has a separation problem. These had never been separated. So we took a look. What we found was quite interesting. You might recall from undergraduate days, if you went to undergraduate school a while ago, back in classic quant class, there was a gravimetric analysis technique where you could use barium ion to precipitate sulfate, so you could determine the amount of sulfate in an unknown sample. These things just precipitated, you filtered it off, you dried it, you weighed it, and you could determine the concentration of sulfate. So barium binds very strongly not only to sulfate, but also any organic sulfonate or any organic phosphate compound. We also happen to know that barium binds a bit to cyclofructans. So the question was, can we add a small amount of barium to the mobile phase and get a separation of all these phosphate and all these sulfonate compounds by adding a little bit of barium, which would bring the two together? And the answer is it worked beautifully, as you can see from the separation on your right. Here is another effect. The first two peaks are a chiral phosphate compound. The second two peaks are a chiral sulfonate compound. They are CPA1 and CPA5, which are both atrope isomers. You can change the retention times and the resolutions by changing the amount of barium, a small amount of barium in the mobile phase. And it worked quite nicely. And for preparative, since he had to prep up on some of these, it's a wonderful preparative solution to things. Here you see the preparative separation of a chiral sulfonate, which is an atrope isomer. We injected 800 microliters of sample at a concentration of 48 milligrams per liter. So in one shot, we got a 99% purity over 20 milligrams of each enantiomer. That was pretty good. For all the prep and semi-prep separations we did, we rarely get that much with one shot, and that purity as well. A couple of years later, the Crochet group approached us again and said they had a really unusual separation problem, and they were having trouble getting any kind. They developed this racemate cage compound with iridium in the middle, and you see a structure on the right here, and they tried all their columns and nothing seemed to work. Did we have a possible solution? Well, we got the structure of this compound just after we finished the work with McFarland on the ruthenium complexes using the cyclofructan RN column. And I said, I think we have something that's going to work for this atrope isomer, essentially, is what it is. And so we went immediately to the cyclofructan RN, and you look in the upper left-hand corner, you see we have a beautiful separation of the M enantiomer from the P. We used a circular dichroism detector on this. You see that trace below. You get a negative peak for the M enantiomer, a positive peak for the P enantiomer. But the really interesting thing we found out when we used a slightly different cyclofructan derivative, the DMP on the right, we reversed the enantioselectivity. Here, the P enantiomer came out first, and the M enantiomer came out second. So that was sort of a surprising find that we didn't expect, and we were able to prep up on both of these things. The other thing I particularly like about this chiral detector, which is the CD2095, is that you can also get the complete electronic CD spectrum for the compound that's coming off your HPLC, and those are shown below. So let's look below A at B. There you see the blue is one enantiomer, the CD spectrum of one enantiomer. The red is the CD spectrum of the other enantiomer. So, and then to use it as a detector, you simply lock in at one wavelength, 320 nanometers, and you get the chromatogram that you see above it. So the fact that you can do that and get the entire electronic CD spectrum, we really like to be able to do, because we use that for a lot of different things. And once again, we got the absolute configuration of these things with Jordan Nafty. Seems like every 2 or 3 years, the Crochet group comes up with separation for us to do that they swear can't be done on any known chiral stationary phase. And this was another one of those cases. This was just about 1.5 years ago. And He was doing an asymmetric synthesis, and on the right here you see the final product. And they said they tried every column, including the cyclofructan RN. Nothing worked. This compound has a molecular weight of almost 1,000. It has a single chiral center right there. So it's buried in the middle of this compound. And fortunately for us, about a year and a half before Mike approached us to do this separation, we developed a new column, which we call the Nicoshell column. This column was named Nicoshell. All these columns, by the way, are core shell. So they give much higher efficiency than the standard fully porous particles. And they give, on a regular HPLC, they give the efficiency of UHPLC. I'll show you some examples of that later. But back to the Nicoshell, we developed this shortly before. And it was named Nicoshell because it does nicotine better than any other column and all the nicotine derivatives, but it turns out it does a whole lot more as well. And so we took the compound, and there you see the separation of the racemate. And on the right, you see we were able to determine the enantiomeric purity of his final product had a good enantiomeric purity of 93%. The other benefit of this unusual mobile phase, which was 10% methanol, 10% ethanol, 80% hexane, is all the impurities in this, which you see here and here, came out before the enantiomeric pair. There's over 20 of them. And it's one of those cases where, you know, there's lots of impurities, and if these things aren't separated from your enantiomer of interest, it's going to be hard actually quantitating the peak areas in this case. But the Nicoshell worked fantastically, and he was able to publish in ACS Catalysis. Now, another organic group that we work with is Carl Lovely's group here at UTA, and he does total synthesis of natural products, and these syntheses get pretty involved very quickly. And so he was doing haplosclerotamine, if I can pronounce it correctly, haplosclerotamine, and it was a 1, 2, 3, 4, 5, 6, 7, 8-step synthesis. The one he finally came up with. It took several trials before he got to this point, and he needed the EE for every step along the way, including the final product. And it turns out, fortunately, we had developed a new Nicoshell column, also a maltose shell column. We were able to use one of those two columns for every enantiomeric excess, for every intermediate, and the final product. You see those shown here. So the top A has an enantiomeric excess of 50%, and over on the other side, another enantiomeric excess of 50%. The racemate separations are shown below. So we were able to get all the intermediate EEs and the final products for the Lovely group. This last project is one we worked on with Merck in Rahway, New Jersey, Merck Pharmaceutical. And Merck had a process that they considered unacceptable. So they were doing an asymmetric synthesis of the entire set of intermediates to get to an Alzheimer's drug. And they needed the EEs of every intermediate and the final product. They had a process when they approached, before they approached us, they worked out a process that they considered unacceptable. It got 4 of the 5 intermediates, but not the 5th, and it got the final product. But it was unacceptable to them for a couple reasons. One, they had to use 2 different separation modes. They had to use SFC and HPLC. What could be separated by HPLC couldn't be separated by SFC, and vice versa. So they had to have 2 systems depending on the column available. They had to use 5 different chiral stationary phases, 4 different mobile phase preparations, including a pH control. And they had to have 5 different methods to be validated and transferred to the manufacturing facilities. This wasn't going to work. What they wanted was to transfer a reverse phase method to the manufacturing facilities that was easy to use and consistent across everything. So we did a screening experiment, and we're going to ignore the upper SFC screening experiment, looked at the reverse phase screening experiment on the bottom. And in this, On the far right side, you see something called the Tico shell. The Tico shell had the best on the screen, had the highest enantioselectivity for all 5 of the precursor compounds, all 5 of the intermediates, and the second best for the final product. But the Nico shell next door had far and away the best enantioselectivity for that final product. So they chose to use Tico shell for all the intermediates and Nico shell for the last. And they were all done in the reverse phase mode. And by the way, here are the structures of the intermediates and the final API. And so now what they transferred, everything was in reverse phase LC. They had only 2 chiral stationary phases. They could all use a conventional HPLC. They didn't have to use a UHPLC. By the way, these are just the screening results. They only needed 2 mobile phase preparations for everything, all 6. And it was easy to validate and transfer to the manufacturing facilities. And that's the method they used, and that's what they transferred. So it was always nice to help out Merck in this case. Now, let's change gears a little bit, and we'll focus more on SFC. We've been doing SFC for 2.5 decades, and methanol is, of course, the accepted modifier, most used modifier for SFC. But we started looking at adding small amounts of water to the methanolic modifier to see what it would do, and we were surprised to find out in many cases things got a lot better. So let's look at what happened? There we go. Okay. So let's look in the upper upper left-hand corner, excuse me. This is a ruthenium compound, chiral ruthenium compound. And in this plot, we're plotting time on the lower right, the percent of water in the modifier, the intensity of the peak. And we see in classic conditions with methanol CO2, you get a nice separation, and it takes over 45 minutes, as you can see right here. If we start adding small amounts of water to the methanolic modifier, the efficiency improves and the retention time decreases. And with 10% water in the methanol, we have doubled the efficiency and decreased the retention time 7-fold. This looks like a huge benefit. And, sorry, we keep jumping between slides. It happens with the other compounds of this group as well. And actually, if you want to do an ultra-fast separation by using water in the mobile phase, over on the right, you can see all of these separations done in under 50 seconds. So the question is this a general phenomenon? Can we do this with other compounds as well? But we went one step further. Well, the answer is yes, it does work for other compounds, but we decided to go one step further. Can we replace methanol or methanol water with azeotropic ethanol as the co-solvent for chiral separations in SFC? And this was published in Green Chemistry because it's a very green method. We know, we've already discussed, methanol is the most polar alcohol that's used as the additive in SFC. When you use higher alcohols like ethanol, isopropanol, it gives you longer retention times and reduced chromatographic performance, which I'll show you in a second. On the other hand, azeotropic ethanol, which is 95.6% ethanol and a few percent of water, it's also known as the 190 proof ethanol commercially. I don't know why they call it, but that's what they call it. When you use this as an SFC modifier, we're going to show it produces lower retention and even higher efficiencies than methanol. And azeotropic ethanol is the greenest solvent compared to methanol. Absolute ethanol is less expensive than methanol. It's less expensive than absolute ethanol or methanol, and it's less expensive and it's easier to recycle because if you're distilling it, it's an azeotrope that has a lower boiling point and it removes water as well. So this is a comparison of a methanol separation, and we're looking at 2 different chiral compounds here. There's the first one, and bupivacaine is the second one. There's the classic SFC separation on top, A and D, for these two things, it's a good separation. It's a nice separation with methanol. If we switch to absolute ethanol and look below it, those two chromatograms, the selectivity is a little better on the left, it's no better on the right, and the peak shapes are much worse with absolute ethanol. But when you go to the 190 proof ethanol on the bottom, what you see is a significant decrease in the retention time, a significant increase in efficiency, a far superior separation, SFC separation. So this may be the way to go, and may be a good replacement for methanol. Here's another example of the same thing. This time we're separating 4 beta blockers, and we're doing all 4 at the same time. Top separation is 190 proof ethanol. By the way, the mobile phase is 80% CO2 with 20% of the ethanol or methanolic modifier. So in green, we have 190 proof ethanol. The middle red chromatogram is the classic methanol additive, and the bottom is the absolute ethanol. Clearly, absolute ethanol is the worst, but 190 proof ethanol is the best, providing the lowest retention and highest efficiency. What about for preparative or semi-preparative separations? How does 190 proof ethanol work there? So let's compare it directly to methanol. This is effexor enantiomers done on a Vanco shell column. And on the left, you see using the methanol modifier. So we're 80% CO2 and 20% modifier. If you look at the very bottom chromatogram, the black curve, that's essentially the analytical separation with 6.25 millimolar sample injected. The minute we increase the concentration to about double that, you get the red curve. You get the red curve, and already the peaks are overlapping. And then the green curve, greater concentration. The top curve, greatest concentration, 50 millimolar. You get a lot of peak overlap. But with 190 proof ethanol, doing the exact same experiment, just substituting the 190 proof ethanol for the methanol, you essentially get baseline separations at all concentrations and a better analytical separation, better peak shape, and with a better peak shape, eliminating the tailing and some of the fronting, you essentially get baseline separation all the way to the highest concentration. So it's really advantageous in many cases to substitute 190 proof ethanol, which is cheaper, easier to recycle than even methanol. Now let's change gears completely, and we're going to look at a whole different set of chiral molecules, and these are alpha hydroxy acids. There's a lot of renewed interest in alpha hydroxy acids as biomarkers, and we're going to be using a tecoshell column for these. And we want rapid, high efficiency separations, and we want to develop a separation using HPLC mass spec and HPLC UV detection. Can we do both? Now, why is there interest in, and the most interested right now is in lactic acid and 2-hydroxyglutaric acid. If you have a short bowel syndrome, this is the lactic acid profile. You have a predominance in your serum of D-lactic acid and not the L-lactic acid. If you are healthy, you have the opposite occurrence. The L-lactic acid is dominant and the D is the minor enantiomer. And this is being used to diagnose hypoxia, sepsis, pancreatitis, all sorts of different things, diabetic ketoacidosis, and so forth. Now, on the right, we have hydroxyglutaric acid, and it is found to be an excellent biomarker for a lot of genetic disorders and for different tumors. So these things are receiving a lot of attention today. This is a very busy slide. We looked at 11 hydroxy acids. They are listed in the first column. The first 2, lactic acid and 2-hydroxyglutaric acid, I think are the most important, but there are 9 more if you're interested. This is the optimum mobile phase conditions for LC mass spec separation. There's baseline resolution for everything. Fine. The one thing you optimize is the organic solvent, it's either acetonitrile or methanol, and you change the ratio of ammonium formate to get the best mass spec and the best separation. So just a couple examples. For lactic acid, you see with 20 millimolar ammonium formate, you get by far and away the best separation. But for phenolactic acid, a close relative, the best separation is 50 millimolar ammonium formate. So you have to change the ratio of the buffer to optimize the separation. That's pretty easy to do. The hydroxyglutaric acid was a very interesting case. Here we did the separation on the left with methanol, and we did the separation on the right with acetonitrile. Both are excellent separations, nice baseline separations. But the interesting thing is the retention time. With acetonitrile, the separation took 38 minutes. With methanol, it was done in under 4.5 minutes. This surprised us, this big a difference. And so obviously, methanol is the way to go. So classically, in classic reverse phase separations, acetonitrile is a stronger additive, organic additive than methanol. But here, methanol elutes things quicker, just as sharp, and gives you a beautiful separation with LC mass spec. Now, let's go back to the structure. If you look at the structure of lactic acid and 2-hydroxyglutaric acid, if you want to do these by UV detection, you have a problem, which is there's no chromophore. There is in phenolactic acid, but it's the only one of the 11 that we had an aromatic group. All the rest of these things have a carbonyl group, and that's it. So in order to see these with UV detection, you're going to have to be 200 nanometers to 205 nanometers. And practically everything absorbs at those wavelengths. So how were we able to get a separation of these compounds using only UV detection? We had to use totally different mobile phase conditions. We had to restrict ourselves to acetonitrile in the mobile phase because it didn't give an appreciable signal at 205 nanometers, and we had to restrict our buffer, our salt component, to potassium dihydrogen phosphate buffer, pH 5, because phosphate doesn't absorb at 205 nanometers appreciably. Using this particular combination, again, we were able to separate all 11 of the hydroxy acids with baseline or better resolution. I'll show you a couple examples of that. Let's first start with mandelic acid over on the far left. So for some of these compounds, you can buy standards, the enantiomeric standards. So if you look at Sigma Aldrich, you can buy R-mandelic acid. This is R-mandelic acid. But lo and behold, R-mandelic acid has some small amount of the S enantiomer in it. So what if you buy the standard, the chiral standard of S-mandelic acid? Well, the S-mandelic acid has some small amount of the R enantiomer in it. We can detect this with a UV detector. In fact, this is true for all hydroxy acids. There are no completely enantiomerically pure alpha hydroxy acids. And by the way, the same thing is true for alpha amino acids. There are no enantiomerically pure D or L amino acids that you can buy. They always have small amounts of the other enantiomer in them. And this shows you an example for alpha hydroxy acids. And over here, the percent of the enantiomeric impurity that we found in each. Okay, now let's really change our direction. This is going to be something that is going to happen in the software of all HPLCs, UHPLCs, and SFCs eventually. And because I'll show you what it can do. And it can give UHPLC efficiencies to ordinary HPLCs. And it also helps with specialized detectors where you have extra column volume, like circular dichroism detectors or polarimetry detectors or evaporative light scattering detectors. These things have big extra column volumes. The trick is, can you remove that extra column volume? So if you look at the empirical chromatogram below, that might be your peak shape at about 5 minutes, but that's not what's coming out of the column. That's what's coming out of the column, plus the extra column band broadening from your detector and so forth. The question is, can you get only what comes out of the column and ignore the detector contribution and get a chromatogram that you see like this that has much higher efficiency? And the answer is you can. It's with the right software, which we have developed. What you do is you take the column out of your system, any system, any regular HPLC, UHPLC with any detector, and you hook your injector with a zero dead volume fitting directly to the detector. You shoot your compound and you'll get a peak. And that peak, there's no column in there, so it's just from the extra column volume of the detector and so forth. And if you have a mathematical equation to describe the shape of that peak, which we now have, you just store that in your computer memory. You then put your column back, and you only have to do that once for your HPLC. Then you put your column back in, shoot it. You get this classic chromatogram. You can deconvolve those and remove the extra column volume to get the higher efficiency column only chromatogram. So does it work? Well, we tested it with UHPLC, a regular HPLC, a circular dichroism detector, and an evaporative light scattering detector. You get the smallest effect with the UHPLC, which you'd expect because the UHPLC has already minimized the extra column volume and extra column effects in the detector. They had to. So you just get a small increase from 17.2 plates to 17.8 plates for UHPLC. So it's not going to give you that much for UHPLC. But what about a regular HPLC? In a regular HPLC, you go from the black trace, which is your original chromatogram, to the blue trace, which is your deconvolved chromatogram. So the regular HPLC, same separation as you did with the UHPLC. You went from 17,200 plates to 14,000 plates. You lost a lot of plates because you're doing regular HPLC. But the deconvolved HPLC has 18,400 plates. You're doing as well as you do with UHPLC for both the first enantiomer and the second enantiomer. So you can get HPLC efficiency or UHPLC efficiencies with an ordinary HPLC using these deconvolution technique, which is quite simple. What about for specialized detectors like circular dichroism detectors? In C, you see a circular dichroism separation. The black is the original detector, and we had only 4,500 plates for that. When we deconvolve it, we go to 12,700 theoretical plates. And for the second peak, we went from 3,800 plates to 8,700 plates. So it's greatly enhancing the efficiency. Same thing is true for evaporative light scattering. We greatly enhance the efficiency that inherently you lose with these larger volume detectors. And these detectors have to be larger volume because they have just a longer path length or a sensitivity problem. They have to do this in order to work properly. So we think this is coming and it's going to be predominant everywhere eventually as the manufacturers put this into their software. And you can just do it with a click of a mouse. So having said that, let me thank my research group. This is the group, a new group picture. These are the people that do all the hard work in the lab so that I can give these presentations. They're quite a illustrious group, and they're doing a great job. And I also need to thank some of our collaborators, Sherry McFarland, Carl Lovely, Eric Rigolato of Merck, Jordan Nafty, and Mike Crochet, who we've done a lot of these co-projects with and solved a lot of difficult separation problems. So thank all of them and thank you for listening. And if there are any questions, I'll be glad to answer them.
Okay, thank you very much. Thank you, Dr. Armstrong, for that very informative presentation. Sorry about that. So for those of you that arrived after the webinar started, you can ask questions by clicking the chat icon at the top of your screen. So looks like we have some questions that have come in already. So the first question is, what CSPs have the broadest applicability or do unexpected separations?
Well, right now, the broadest chiral stationary phases are the Tico shell and the Vanco shell. And the Nico shell, which is the most recent one, is doing, if nothing else works, we try the Nico shell. For the cellulose polymeric, we like the IC, or we like the IA and the IC. They're more normal phase SFC type stationary phase. But all the Tico shell, the Nico shell, the Vanco shell, they have just as broad a selectivity. They're just used with different mobile phases, usually polar organic mobile phases and or reverse phase mobile phase. So they both have broad selectivities, but usually different mobile phases are used for those two classes of stationary phases. The Tico shell, Nico shell variety are all on core shell columns, so you're going to get higher efficiency because they're on a core shell particle. And we've just added the new Welco shell, by the way, to that on a core shell particle. So that was a nice addition. I didn't have a chance to talk about any of those separations because we're just getting ready to publish them.
Okay, thanks. So next question. What CSPs are superior for non-enantiomeric separations?
This is interesting. Of course, people think chiral stationary phases are only for chiral analytes, but over the last 10 or 12 years, it's been found that you can use a chiral stationary phase and get a better separation for non-chiral analytes than you can with a traditional non-chiral stationary phase. And there's a couple groups of molecules that this is almost universally true for. One is peptides. So if you're doing short peptides to medium length peptides, you can separate peptides, many of them on a C8 or C18 or C4 stationary phase, but on a chiral phase, in the reverse phase mode, you usually get higher selectivity. You can separate a greater number of these peptides and usually with far different selectivities. So a lot of people use, for peptides, use chiral stationary phases. Another group of molecules, this also came out of Merck, by the way. We're talking about fluorinated pharmaceutical compounds. So the biggest impurity when you have a compound with fluorine somewhere in the structure, the biggest impurity that follows everything through the entire synthesis is the desfluoro analog, where the fluorine is popped off and it's replaced by a hydrogen. Separating the fluorinated compound from the desfluorinated impurity is sometimes more difficult than a chiral separation because the hydrogen and the fluorine are about the same volume on the structure. And so both we and Merck have done a series of papers showing the separation of fluoro and desfluoro compounds are better done on chiral phases than on achiral phases. So that's at least two examples, but there are others out there. I mean, the chiral phases have all these additional interactions that you can do that allow you to achieve a lot of achiral separations with greater selectivity and sometimes greater speed.
Okay, next up, what are the best green chiral separation methods?
Boy, this is coming. I actually had a couple of slides at the end of this presentation. Can I go to slides after this?
Yeah, I believe so.
Okay. I took these out because of time limitations. We just published, this is the cover of Green Chemistry, just came out a month ago. Okay. And it's emphasizing the analytical method greenness score of the AMG. You can actually calculate the analytical method greenness using an equation that we present that was previously developed in simplified form by the American Chemical Society. And in this case, we're looking at water and CO2. The two greenest solvents are water and carbon dioxide. And so if you can limit yourself to those two solvents, you're about as green as you're going to get. So what can you do with water and carbon dioxide, which is essentially carbonated water? Oh, let me backtrack a bit. The analytical method greenness score allow you to compare methods that have been published in the past with new papers that come out that claim to be green or sustainable. What we're saying is you can't always believe these green sustainability claims. This is a figure from the paper. Look at method 3. Method 3, shown here in the middle, the greenness score, the AMGS score, is the size of these bars in the bar graph, and the different colors, red, green, blue, are the different contributions to this: health and safety, instrument powers, how much does it cost to dispose of a solvent and make a solvent, things like this. So this came out as a sustainable method because it used ethanol. So we calculated the AMGS score for this. Then we looked at a paper for the same amino acids, KFTV amino acids. The same amino acid was published 26 years ago when nobody cared about greenness. And the method from 26 years ago was greener than the sustainable method that they just published. So just be aware of this. This figure also shows you that supercritical fluid separations tend to be greener than HPLC separations. These first two are the supercritical fluid separations. At any rate, we wanted to go as green as you could go. We used amino acids as the test molecule. Can you do separations with just water, CO2, carbonic acid? We looked at 38 amino acids, including all 19 of the chiral proteogenic amino acids. You can do half of them baseline with carbonic acid, CO2 and water, nothing else. And you can get essentially all of them with one exception, which I have to explain perhaps later, by adding a little bit of methanol. So that's about as green as you can go. And it turns out that this particular column is going to be on the Mars lander. It's going to be the first HPLC column. It's a tecoplanin column. It'll be the first HPLC column on an extraterrestrial planet, as it'll go to Mars and it'll be analyzing amino acids, hydroxy acids, other things. And they don't want to be hauling a lot of organic solvents up there. They want to use carbon dioxide, which is the atmosphere of Mars, and they'll bring up a little bit of water. So hope that answers that question, at least in part. I think we're going to go a lot towards these greener separations, and it's going to have an impact, big impact on the pharmaceutical industry.
Interesting. Okay, so we have time for one more question. How useful has the CD detector been for chiral separations?
We use it all the time. First of all, to determine initially, is there actually a chiral separation? I mean, I would suppose in 30–40% of the cases where we first start doing a chiral separation, we get two peaks, and one peak's the racemate, and the other peak is some impurity. And so, yeah, you can tell that right away if you have a CD detector, because you get no signal for either one, unless the impurity happens to be chiral, but you certainly won't get a signal for the racemate. And it also really helps when you're reversing elution order. You want to determine if you reverse elution order for two enantiomers, which frequently happens. And if you don't know it's happening, that could be problematic. So we almost always use it in line, sometimes separately from our UV detector, and sometimes we use it to get the entire CD spectrum, because there are cases where you can determine the absolute configuration from the electronic CD spectrum rather than vibrational circular dichroism. So we have used it for that as well.
Okay, so that looks like it concludes our messages, our questions. If you did not get a chance to ask your question, please email the address that you see on the screen, sales@jascoinc.com. We will be taking them and forwarding them over to Dr. Armstrong. So again, Dr. Armstrong, thank you for that very informative presentation, and I hope everybody has a great afternoon.
Okay. Thank you.